Okay, Assalamualaikum and a very good morning to everyone. Welcome to the first biology class. Okay, so as I have mentioned to you a few days ago that we are going to cover the missing subtopic from a few topic that you actually should learn. Okay, in Form 4 KSSM. Okay, now without further ado, we are going to continue our lesson. Okay, so let me set up first. Okay, now we are going to study the first one, which is about the cell structure and organization. Okay, so okay, okay, good. Okay, guys. So for the first one, we are going to study about unicellular organism. Okay, so there are two examples of unicellular organism you should know. The first one is amoeba, so it will look like this. Okay, and the next one is we have paramecium. Okay, so it will look like this. Okay, so let me make it larger. Okay, guys, so for the first one, we should learn about the structure of amoeba. Okay, so this is how amoeba looks like. So basically, this amoeba, its habitat is in the freshwater pond and also damp soil. Okay, so this is the general characteristic of amoeba. The first one, it can change the shape constantly. Okay, so obviously you see here there is no cell wall, so that's why amoeba cannot have a fixed shape, so that's why it can change the shape, okay, constantly. And it has two types of cell wall. The first one is ectoplasm. Okay, ectoplasm is the outermost layer, and then they also have endoplasm. Okay, so endoplasm is the one that in the middle. Okay, so this is ectoplasm and this is ectoplasm. And then they basically feed by using pseudopodia. Okay, so as you can see, this is the amoeba. So what does pseudopodia means is that, okay, so this is pseudopodia means. Pseudopodia means like the extended structure, okay, made by the amoeba. Okay, so basically they will just be like this, but if they want to feed, okay, they will form pseudopodia. So here is pseudopodia, where it is a finger-like structure. Okay, so after this we are going to study in detail. Okay, guys, so these are the process that we should learn about amoeba. The first one is locomotion. Okay, and then feeding, respiration, excretion, response to the stimuli, and the last one is reproduction. Okay, so we go for the first one, locomotion. So as I said just now, they have pseudopodia structure, right? Okay, so not just for feeding, in locomotion also, pseudopodia is needed. Okay, so you see here, pseudopodia are used to anchor, okay, the tips of the ground, and followed by the flow of cytoplasm into pseudopodia, and the form of the locomotion is known as a amoeboid movement. Okay, so this is the structure showing to you how amoeba move. The first one, okay, this is the general shape of the amoeba. So when it want to move forward, what it will do is that it will form pseudopodia. As I said just now, pseudopodia is a finger-like structure. Okay, it is a extended, okay, finger-like structure like this. So here, this is amoeba. Amoeba will form pseudopodia, so it will have this structure. So what will happen is that the cytoplasm, okay, in the pseudopodia, the cytoplasm will flow to the pseudopodia. Because here, this is pseudopodia, so if it want to move forward, so the cytoplasm, okay, will move towards, okay, pseudopodia. So this will enable the amoeba to move forward. Okay, so for example, if the amoeba want to move left, so it will form pseudopodia to the left. So what will happen next is that the cytoplasm will move towards the pseudopodia. So sooner or later it can move forward. Okay, and then bear in mind the function of pseudopodia is also to anchor. Okay, anchor tu maksudnya mencengkam. Okay, the tips of the ground. Okay. Okay, that's all for the locomotion. The next one is in terms of feeding. Okay, so the name of the process where the amoeba feed is known as a phagocytosis. Okay, so actually is quite the same like when they want to move. So what will happen first is that, okay, amoeba will form a pseudopodia. Why? Because they want to engulf. This you look this. This look like a finger structure, right? So what will happen is that two pseudopodia is formed to engulf. Engulf tu maksudnya to surround pun boleh. Okay, to surround the food. Right now we know that amoeba feed on bacteria. Okay, so what will happen is that, okay, once the pseudopodia, is formed and they engulf the bacteria, they will form food vacuole. So here is food vacuole that contain bacteria. So what will happen next is that the digestive enzyme will break down the food and the nutrient diffuse into the cytoplasm. So here we have food vacuole. Okay, and then what will happen is that the digestive enzyme, okay, will move inside the food vacuole, because we know that in order to get the nutrient from the food, we need to digest using enzyme. So once the bacteria is digested, any nutrient, okay, available, okay, will diffuse into the cytoplasm. Okay. Okay, enough with the feeding. Next is respiration. Okay, so here it say that respiration in amoeba, okay, occur by diffusion, okay, or simple diffusion, where the oxygen can easily diffuse in and carbon dioxide can easily diffuse out. So it is not like a human. Like for us, if we want to take oxygen, we will breathe using nose. And then the oxygen will go to the lung. And then from lung, it will go to all over the body, right? But for amoeba, because it is a unicellular organism, it use simple diffusion where the oxygen and carbon dioxide can diffuse in and out easily, just like that, through plasma membrane. Because here we know that the outermost layer is known as a plasma membrane. Okay, it's very straightforward information. And the next one is excretion. Okay, so basically excretion, we know that is the process to remove the waste product. Okay, so the first one is carbon dioxide, because we have learned just now, carbon dioxide can easily, okay, diffuse out from amoeba using simple diffusion, where the carbon dioxide can diffuse just as easy as that. Okay, at the same time, for the amoeba, they have contractile vacuole. Okay, so this is what we call contractile vacuole. Okay, the function is to remove excess water in the cell or in the amoeba. Okay, so here I want to explain in detail. Okay, just now we know that amoeba basically they live in the fresh water, right? So when they live in the fresh water, it means that they are constantly absorbing the water. So we can say that there are so many excess water will diffuse inside the amoeba. So how can the amoeba remove the excess water? So what will happen is that the excess water will diffuse into contractile vacuole. Okay, so when the so many water in the amoeba, the water will diffuse in the contractile vacuole. Once the contractile vacuole is full of water, okay, so this contractile vacuole will merge, okay, with the plasma membrane to what? To eliminate the excess water. Okay, so that's why the contractile, oh sorry, that's why the function of contractile vacuole is to remove excess water in the cell, in the amoeba lah. Okay, the next one is reproduction. There are two conditions, that need to be faced by amoeba. The first one is under favorable condition. So under favorable condition, basically they will do asexual reproduction, which is binary fission. But when they are under unfavorable condition, they will do sporulation. Okay, and this is sexual reproduction. Okay, so you might be wondering what kind of condition for favorable. Okay, for example, favorable is a condition like, you know, during a good day when there is sunshine, there is enough water, and the humidity is stable. Okay, that one is for favorable condition. But unfavorable condition, for example, during drought. Okay, during kemarau, for example, okay, for drought, during whatever lah, that they have to bear. Okay, so during favorable condition, they will do binary fission, but unfavorable, they do sporulation. Okay, so how it works? So we go for favorable condition first. Okay, binary fission. Okay, so here, okay, during favorable condition, how it can reproduce is that the amoeba will divide after it has grown into a certain size. So it means that once the amoeba has grown up, we can say that it is ready for reproduction. So the first one, the nucleus, okay, the nucleus will divide. Okay, from one it become two. So once it has two nucleus, then it will divide the cytoplasm. Okay, the cytoplasm begins to divide when the nucleus has split up. So, as you can see here, after division of cytoplasm, two daughter amoeba are formed. Okay, so this is what we call binary fission. Okay, this one is under unfavorable condition. So when the environment is not favorable, okay, so the first one, this is amoeba. Okay, so the amoeba will form a cyst. Will form a cyst, the outer layer of many spore that contain many spore. Okay, so at the thermos layer of the amoeba, it will form cyst. So inside the cyst, there are so many spore. And then the spore will be dispersed by the wind. Okay, the cyst germinate and they release the spore, and the spore will be carried, okay, by the wind. Okay, so the wind will carry the spore to the other destination that might be favorable for them. Okay. And then the next one is, okay, the last one, growth. Okay, so we know that amoeba will grow by synthesizing, okay, a new cytoplasm. Okay, so it's very straightforward information. Okay, the next one. Ada bunyi pula. Okay, the next one is level of organization. Okay, so this one I think you are familiar. Okay, we start with cell, tissue, organ, system, and also organism. Okay, this one I think is very straightforward information. So this one's, okay, we begin with the cell. Okay, cell is the smallest unit of life capable of carrying out all the function of living thing. So there are two types of cell, plant and also animal cell. Okay, so this is the example of cell. Okay, nerve cell, okay, blood cell, sperm cell, and so on. Okay, you should know, okay, the function of each cell. For example, sperm cell, the function is to what? Okay, I think this one is very important for you to know yourself. Okay, okay, so this is the types of blood. Okay. Okay, the next one is tissue. Okay, so there are 4 types of tissue that you should know. The first one is epithelial tissue. Okay, we go for the first one. Okay, epithelial tissue consists of cells that are arranged tightly. Okay, and they could be found, okay, in a body surface, lining of cavity, and also gland. So I make it simple. I make it organized for you. So there are 3 places where you can find epithelial tissue. The first one is body surface. Okay, so the example is skin. Okay, and the function of our skin is to protect against bacteria. Okay, so one of the location of epithelial tissue is our skin. Okay, the next one is lining of cavity. Okay, basically lining of cavity involve any organs. Okay, for example, lung. Okay, the function is to produce mucus, okay, to destroy bacteria. Okay, other than lung, maybe there is also epithelial tissue in the esophagus, in our respiratory tract and so on. And the last one, we can find epithelial tissue in the gland. Okay, for example, sweat gland. Okay, sweat gland, the function is also to, this one for sweat, the function not to produce mucus. Instead, the function of sweat gland is to produce sweat, okay, to destroy bacteria. Okay. The next one is muscle tissue. Okay, so this is the abundant tissue in our body. So it means that we have many muscle tissue in our body. So there are 3 types of muscle tissue. Okay, so I make it a table for you as well. So the first one is smooth tissue. Okay, here smooth tissue you can find in a in the small intestine. So this is the way how it looks. Okay, smooth tissue. And then the function is okay, responsible for involuntary body activity. For example, we know that small intestine is the place where the food will move, right? So it will, when we have smooth muscle tissue, it will enable the food, okay, to move along our small intestine. And then we have skeletal tissue. Okay, for example, in the leg. Okay, skeletal means skeletal tissue, muscle tissue means that any muscle that is attached to your bones. Okay, so for example here. Okay, so this is how skeletal muscle looks like. It look very long and also tight to each other. Okay, so the function is responsible for voluntary action. For example, walking. Voluntary means that you are aware that you are doing that activity. Okay, for example, it's walking. And the last one is cardiac muscle tissue. Cardiac muscle tissue can only be found in one place, which is heart. Okay, so this is how it looks like. Okay, the function is to responsible for involuntary body activity. Okay, the example is heartbeat. Okay, so because the heartbeat, okay, heartbeat is involuntary. Okay, why? Because we cannot control. Okay, it happen naturally. Okay, the beat of heart happen naturally without our knowing. Okay. Okay, guys. So the next one is nerve tissue. Okay, so this is nerve tissue looks like. Okay, so nerve tissue is made up by nerve cell, and nerve cell is also known as a neuron. Okay, the function is to transmit impulse from receptor to the brain. Okay, and nerve cell consists of dendrite, okay, axon, and also body cell. Okay, body cell. Okay, guys. So the last one is connective tissue. There are 3 types of connective tissue that you should know. We have dense fibrous connective tissue. Okay, so it helps to connect, okay, muscle to the bones. Okay, and then we have cartilage connective tissue, okay, to support our nose and ears. Okay, so right now, can you touch your nose and ears? So it has cartilage connective tissue. Okay. And we also have adipose connective tissue, the place where the excessive fat is stored. Okay, so if you want to know where you can find connective tissue, okay, for example, under your arms, okay, lengan awak tu. Okay, under your arm, there is a adipose connective tissue. Okay, why? Because that is the place where any excessive fat will be stored. Okay, so that's why you see, okay, if you want to see either you are fat or not, you see under your arms. You can see at your thigh, okay, dekat peha awak tu. Peha can also have adipose tissue. Okay. Okay, the next one is organ. So I think organ is very easy for you. You know, heart, okay, lung, and so on. But for plant, okay, the example of their organs is leaf. Okay, leaf, stem, flower, and so on. So system is very straightforward, I think. Dia bising lah pula. Sorry. Okay, so these are the example of system. Okay, that you can find in your body. I think this one is very straightforward. You can read on your own. Okay, so there are a lot of system. Okay, exist in our body. Okay, skeletal system, circulatory system. Okay, now moving forward to the plant. Okay, plant there are three tissue. The first one is epidermis tissue, ground tissue, and vascular tissue. Okay, so the first one is epidermis tissue. So epidermis tissue you can find in the leaf, stem, and also root. Okay, so here. Okay, this is epidermis tissue. Okay, you can find in the leaf. Okay, so this is the example of leaf. Okay, the next one, ground tissue. There are three types of ground tissue, which are parenchyma tissue. Okay, this is how parenchyma tissue looks like. Okay, so the function is to store the product of photosynthesis, such as starch. So when the plant finish with photosynthesis, it will store the starch in parenchyma tissue. Okay, and the next one is collenchyma tissue. Okay, so you see the difference between these two. Okay, but the function is different. Okay, to give support to herbaceous plant. Okay, and the last one is sclerenchyma tissue. Okay, the function is still the same. Okay, to support and strengthen the plant body. Okay, that one is for tissue. The next one is vascular tissue. Okay, there are two types of vascular tissue, xylem and phloem. I think this one is very familiar to you. Okay, we have transport water. We have xylem tissue and phloem tissue. Okay, so here phloem, this is xylem. Okay, so there are two systems in the plant, shoot and root. Okay, here you can see. Shoot system consists of flower, leaf, fruit, and so on. But for the root system, only root. Okay. Okay, guys. So that's all for chapter 2. Now we are going to move to the next one. Okay, let me see. Okay, the next one is this chapter. Okay, the movement of substance across the plasma membrane. Okay, guys. So, okay, this is how plasma membrane looks like. Okay, okay, I make it bigger. Okay, so this is how plasma membrane looks like. It is also represent as a fluid mosaic model. Okay, here. Okay, so plasma membrane consists of phospholipid and also protein. So this is plasma membrane as a whole. It consists of protein. Okay, the blue color is protein. Okay, and then here, okay, the yellow one, and also in the middle, we know them as a phospholipid. Okay, phospholipid. So plasma membrane consists of phospholipid and protein. Okay, phospholipid and also protein. And then phospholipid bilayer are arranged in two layers. That's why it is called phospholipid bilayer. Bi means two. Okay, and then there are a protein dispersed throughout the phospholipid bilayer. So there are so many protein dispersed. Okay, throughout the phospholipid bilayer. Next. Okay, protein found in the phospholipid bilayer. So basically, there are a few types of protein you can find in plasma membrane. The first one is receptor protein. We have channel protein, and it is always open like this. Then we have transport protein. We have gate channel protein. And the last one, we have glycoprotein. Okay, glycoprotein is very special because it's combination between carbohydrate and also protein. So this is protein, this is carbohydrate. So as a group, we call them as a glycoprotein lah. Other than that, in the plasma membrane, there is also cholesterol. So here is the cholesterol. Okay, the function of cholesterol is to strengthen, to strengthen the structure of phospholipid bilayer. Okay, as you know, phospholipid bilayer should be firm, should be stable. Why? Because there are so many substances that will move in and move out from the cell. Okay, so this is the function of the protein that I have mentioned just now. Okay, we have channel protein and we have carrier protein. The function is to transport substance in and out of the cell. For example, glucose, amino acid, and so on. And we have also glycoprotein, as I said just now. This one, glycoprotein. There is a carbohydrate attached on the protein. Now we call them as a glycoprotein. Okay, and then we have receptor protein for cell recognition. Okay, this is for cell recognition because we know that we have so many kind of cell, right? We have red blood cell, we have sperm cell, and so on. So every single cell has different receptor protein. That's why I say it is for cell recognition. So that's it for protein. Okay, in terms of phospholipid bilayer, because we know that plasma membrane has two. The first one is protein, the next one is phospholipid bilayer. So it has two things. The first one is polar hydrophilic head, and the next one is non-polar hydrophobic tail. Okay, so this is how it looks like. This is head. Head means kepala dia lah. Okay, this is tail. Okay, we go one by one. Okay, polar hydrophilic tail. Okay, so this is polar hydrophilic tail, and in the middle, we call them as a non-polar hydrophobic tail. Okay, what does polar and hydrophilic means? Okay, I go for the hydrophilic means first. Hydrophilic means that this head, it is attracted. It can attract water. Okay, it can attract water. Okay, but for tail, it is hydrophobic. Hydrophobic, it is not attracted to the water, but it is attracted to the fat. Okay, hydrophilic attracted to the water, hydrophobic attracted to the fat. Okay, and then polar means that the direction, it has direction. For example, the head is facing outside the cell. For this one, the head is facing inside of the cell. Okay, but for tail, it is non-polar because the direction is still the same, which is in the middle, no direction. Okay, here you see the characteristic of phospholipid bilayer, it has two things, head and also tail. Head is polar. Why? Because it faces outside and inside lah. Okay, it is attracted to the water and it is away from the water, but it is attracted to the fat. Okay, okay, facing outside, facing inside. Okay, the next one is about the permeability. Okay, before we want to study about this, you should know what does permeability means. Okay, so permeability of membrane is the ability to enable the passage of all molecule across the membrane. Okay, so in a simple definition, I could say that permeability is ability to allow all things, okay, all thing get in or get out from the cell. Okay, but plasma membrane is not fully permeable. So we can say that plasma membrane is semi or selective permeable. Why? Because it only allows some or certain substance can move in or move out across plasma membrane. It means that not everything can get in or get out from the cell. Okay, so the next one you should know, okay, because we learned just now that uh, plasma membrane has two things, right? Phospholipid bilayer or protein. So this is the example of a substance that we'll use either phospholipid bilayer or protein to get in or get out. Okay, for example, phospholipid bilayer, these are the substance that we'll use it. For example, lipid molecules such as fatty acid and glycerol, gas molecule, oxygen and carbon dioxide, and the last one is water. But for protein, just now we know that we have channel protein, transport protein. They basically will use glucose, amino acid, and also iron. Okay, so this substance will use protein to get in or get out from the cell. Okay, guys, so let's move on. Okay, now we are going to move to the last part of our lecture today. Okay, now we are going to enter the new one. Okay, the importance of water in the cell. Okay, the first one, you should know that water is the most abundant component of organism. Same like us. Okay, we have 80% of our body contain water. And then it is excellent solvent to dissolve sugar or whatever. Okay, pelarut lah, we can say that water can dissolve anything, for example salt, sugar, and so on. And then we use it as a transport medium. Okay, our transport medium is a blood, right? So blood consists of water, okay, which is 90%. And it also can act as a lubricant, okay, to assist the movement of food in digestive tract. Okay, sebab itulah lepas makan kena mi apa? Minum. Gitu. Okay, bye. Okay, and the last one, it helps to maintain the constant body temperature. Okay, for example, when your body temperature is too high, okay, you will sweat. Bila sweat keluar apa? Keluar air ke keluar udara? Mestilah keluar air kan. So that's why okay, water can be used, okay, to maintain the constant body temperature. Oh, ada lagi rupanya. Okay, the last one, to take part in chemical reactions such as breaking down protein and carbohydrate. So we can also use water to, to digest the food. Okay, now moving forward, the characteristic of the water. The first one, in terms of polarity. Okay, as you know that water is also known as the H2O. Okay, it has element hydrogen and also element oxygen. Okay, so it also say that water molecule are a polar molecule because share electron between oxygen and hydrogen will be attracted towards oxygen, which is more electronegative. So you can see here, this is one molecule of water, this is another one molecule of water. Okay, so they are attracted one molecule to another molecule using hydrogen bond. Okay, the polarity produce hydrogen bond and allow water to act as a universal solvent. Okay, universal solvent properties of water allow to universal solvent means that anything can be dissolved in the water. Okay, such as glucose, electrolyte, and so on. Okay, so this is in terms of its polarity. Okay, the next one in terms of cohesion force and adhesion force. Okay, so when there is a water and another water molecule, there will be a force between them, and that is called as a cohesion force. Okay, cohesion force is a force between one water molecule with another one water molecule. Okay, okay, so cohesive force. And then there is also another force which is called as adhesive force. Okay, adhesive force is a force between water and the other surface. For example, the xylem wall. Okay, and this force produce capillary action, which allow water to enter and move along narrow space, such as xylem tube. Okay, xylem tube we can find in the plant. Okay, because xylem we know that the function is to transport the water. Okay, specifically these two force will exist. Okay, in the plant. Okay, the next one in terms of specific heat capacity. So water has high specific heat capacity, which is 4.2 kJ over kg centimeter. Degree Celsius. So it means that we need 4.2 kJ energy to increase the temperature of water by 1 degree Celsius. Okay, so the water absorb a lot of heat energy with a small rise in the temperature. Okay, this characteristic is very important to maintain body temperature of organism. I think it's very easy. You can read on your own. Okay, the next one is enzyme. Okay, enzyme is a biological catalyst that speed up the rate of reaction that can convert the substrate to the product. So there are so many enzyme. Okay, you should know. For example, amylase. Okay, so the amylase will speed up the digestion of starch into maltose. I think it's very easy, right? I think you should know this. Okay, so this is the general characteristic of enzyme. The first one, enzyme is made up by protein, and then enzyme can speed up the rate of reaction. So if we use enzyme, we can speed up the conversion of starch into maltose. Okay, and then enzyme has a special structure called active site. Okay, active site, it will look like this. Okay, so for example, this is enzyme. Okay, so here, this is the place. Okay, like I highlight here. This is the place where the substrate, will combine with the enzyme. So this place is known as an active site. Okay, the efficiency of enzyme is lowered by inhibitor. Okay, when there is a presence of inhibitor, this will slow down the efficiency of the enzyme. And then the enzyme is needed in a small amount because they are highly efficient to speed up the reaction. So we don't have to waste the amount of enzyme because it can highly be efficient to speed up the reaction. An enzyme can speed up a forward and reverse reaction. Okay. And the last one, enzyme require cofactor to help to speed up the reaction. Cofactor, for example, vitamin, okay, vitamin K, vitamin E, or so on, depending on what digestion we are dealing with. Okay. So how you can name the enzyme? Basically, you just add SASE, okay, at the end of the words. For example, lipase, sucrose, sucrase, maltose, maltase. Okay, so in order for you to easy to recognize the enzyme, if you see at the end of the words, if there is ASE, it means that it is enzyme. Okay, there are two types of enzyme, okay, which are intracellular and extracellular. Okay, what does it mean? Okay, intracellular is an enzyme that produced inside and used inside. Intracellular enzyme means the enzyme is produced in the cell and also used in the cell. Okay, so this enzyme can be easily found in the cytoplasm. How about extracellular enzyme? Okay, extracellular enzyme is produced inside of the cell, but it is used outside the cell. Okay, I repeat, extracellular enzyme is enzyme produced inside the cell, but used outside the cell. Okay, for example, okay, pancreas produce enzyme. For example, lipase, amylase, trypsin. But where will this enzyme will be used? In the duodenum. So pancreas and duodenum are two different locations. Okay, so how extracellular enzyme is produced? Okay, so here how it happened. Okay, we go by number. Okay, so number one, we know that this is rough endoplasmic reticulum. Okay, rough endoplasmic reticulum contain ribosome. So ribosome will produce protein. So what will happen is that the protein synthesized by ribosome enter the lumen of rough endoplasmic reticulum and transported through it. Okay, so this is rough endoplasmic reticulum. Okay, once it reach the end of rough endoplasmic reticulum, the membrane butt off to form transport vesicle. Number 3 here. So once the protein is produced by ribosome, the rough endoplasmic reticulum will pack. Okay, will pack the protein. Okay, and transfer the protein using transport vesicle. Using transport vesicle. So the transport vesicle that contain protein move to the Golgi apparatus and fuse with it. So this is transport vesicle that contain protein. It will go to the Golgi apparatus. Why? Because we know that the function of Golgi apparatus is to modify, pack, and also transfer. For example, you have protein, you want to make enzyme. If you have protein, you want to make antibody. So this is the place, Golgi apparatus. It will change, okay, the protein. For example, awak ada tepung kan, awak nak buat kek ke, awak nak buat kuih ke. So it's up to the Golgi apparatus. Okay, in the Golgi apparatus, okay, the protein are modified into the enzyme and secreted in the secretory vesicle that are formed at the tip of the Golgi apparatus. So once the protein reach the Golgi apparatus will modify the protein into enzyme. So once the enzyme is ready, the enzyme is packed in the secretory vesicle. Why? Because we know that this enzyme will be used outside the cell. So what will happen is that this secretory vesicle will fuse with plasma membrane to release protein. To release enzyme. So now enzyme will go to the next destination, okay, where it can be used outside. Okay, so here is the this is the procedure where we can produce extracellular enzyme. It's just the same like this one, but this is my own note, okay, for you to understand better. Okay, guys, so I think that's all for our class today. So, once you finish watching this video, I hope you can write down your name and your class, okay, in the section below, so that I can take it as a attendance. And then I will distribute to you an exercise and try to complete, okay, because the next class we are going to discuss the answer. Okay, so I think that's all for today. So I hope you can understand. If you have any question, you can PM me. Okay, that's all for today. So, Assalamualaikum. Bye bye.